Hereby, I open this academic ceremony in which Ghalib Mobaraki will defend the academic thesis Inside Cancer Pathology, Human Polyomavirus, and Bovine Meat and Milk Factors. May I invite you to present a summary of your studies and conclusions of your thesis.
Thank you. Dear Prorector, dear the members of the Corona, dear supervisors, dear ladies and gentlemen, dear friends, family, and colleagues, welcome to Inside Cancer Pathology, Human Polyomavirus, and Bovine Meat and Milk Factor. In the next 15 minutes, I'll give a brief summary of my main studies, finding, and conclusion. Cancer is the second most common cause of death worldwide. The number of cancer deaths is increasing as the world population is growing and aging. 80% of cancer can be caused by non-infectious agents such as alcohol consumption, radiation, and smoking, while the other 20% of cancer can be caused with infectious agents, 15 of them by viruses and the rest by bacteria. And recently, other infectious agents named bovine meat and milk factor been associated with human cancer. Currently, 8 known human tumor viruses which contribute to human cancer either by direct or indirect tumorigenesis. As shown in this slide, 3 of these human tumor viruses can cause human cancer by indirect tumorigenesis, by the rest 5 by direct tumorigenesis. My main objective since 1964 to 2008, and only those human tumor viruses at the top of the mountains, and only one polyomavirus, which named Merkel cell polyomavirus, and it's associated with Merkel cell carcinoma in 80% of cases, and other recent infectious agents. This is raised a questions: what other polyomaviruses and infectious agents are potentially associated with human cancer? Because it's important to identify human tumor virus or other infectious agents in order to develop novel approaches in cancer prevention and therapy. In our lab, we screen for these infectious agents by using different molecular techniques in different human cancer. What are human polyomaviruses? They are non-enveloped, small, circular, and double-stranded DNA genome. Nowadays, 12 human polyomaviruses have been known and discovered in the last 2 decades. Based on this timeline, since 1971, the first 2 human polyomaviruses was named Beta polyomavirus and GC polyomavirus, and then it took time till 2007 to discover 2 others, human polyomaviruses, and that's because the development and the improvement of this detection method. And in 2023, last month, another new human polyomavirus named human polyomavirus 16, discovered by Carolina Torres. Among these human polyomaviruses, only one polyomavirus is considered as human cancer, as human tumor, named Merkel cell polyomavirus, as I mentioned previously. My research question and respective studies. Chapter 2, review of the emerging role of human polyomavirus 6 and 7 in human cancer. Chapter 3, we're looking for the reactivation of Bika virus in urothelial cell carcinoma of bladder. Chapter 4, the presence of other human polyomaviruses in urothelial cell carcinoma of bladder. Chapter 5, we characterize human polyomavirus in the cholangiocarcinomas patient. Chapter 6, mapping the human polyomaviruses in renal cell carcinoma. And finally, Chapter 7, we detect the bovine meat and milk factor in renal cell carcinoma tissues. Since human polyomavirus 6 and 7 isolated from skin in 2010 and shared 68 sequence identity, we aim to review those human polyomavirus if they are a possible candidate for human tumorigenesis. We conclude that high prevalence of both human polyomavirus 6 and 7 increase with the age. As well, we conclude high prevalence of human polyomavirus 6 and human polyomavirus 7 in malignant tissue and low in non-malignant tissue. And finally, human polyomavirus 6 was frequently detected in skin cancer, while human polyomavirus 7 was frequently detected in non-skin cancer. In Chapter 3, we looking for the reactivation of Bika virus in the urothelial cell carcinoma of the bladder. Bika virus is suspected as a cause of urothelial cell carcinoma in immunocompromised patients. Here we aim not only to assess the relation of Bika virus positive urine cytology decoy with the urothelial cell carcinoma, but also to evaluate the relation of Bika virus with a different treatment type of urothelial cell carcinoma. What are decoy cells? They are urothelial epithelial cells infected with a virus. In our lab, 22,867 urine cytology specimens were diagnosed for the presence of the decoy cells, and we report 30 patients, and among these 30 patients, 14 patients were diagnosed with the urothelial cell carcinoma, and 16 patients were without. These 14 urothelial carcinoma patients were screened for the Bika presence in the urine sediment and also in the formalin fixed paraffin embedded tissue by using immunohistochemistry and PCR, and they were positive in the urine sediment, but they are negative in the tissue. We also screened the other 16 non-urothelial carcinoma patients for the presence of Bika virus in the urine sediment by immunohistochemistry, and they were all positive. We conclude that Bika virus is unlikely to be involved in the oncogenesis of urothelial carcinoma of immunocompetent patients. We also conclude that Bika reactivation is not restricted to immunosuppression patients. However, the intravascular treatment could be associated with the reactivation of the latent Bika virus. Chapter 4, we go furthermore for the detection of other human polyomaviruses in the same cohort. And we found that Merkel cell polyomavirus was reported in 14%, while GISA virus was reported in 20%. However, we couldn't find human polyomavirus 6 or 7. In Chapter 5, we characterize for the presence of human polyomavirus in cholangiocarcinoma patient. What is cholangiocarcinoma? It is a rare cancer of bile duct, and recently a report from Hong Kong University reported the presence of this human polyomavirus 6 in bile fluid. Our aim to detect this human polyomavirus in the cholangiocarcinoma patient by using different molecular technique in single cell level. 42 CCA tissues were screened by PCR, either degenerated or specific PCR. Based on this Venn diagrams, we found that human polyomavirus 7 was detected 29 out of 42, while Merkel cell was 10 out of 42, and human polyomavirus 6 was 6 out of 42. We also test for the single cell level by using fluorescent in situ hybridization, RNA in situ hybridization, and immunohistochemistry, so not only from the DNA level, but also for the protein level. Our results show some results for this presence of human polyomaviruses in tissues, not only in the bile duct, but also in the liver cell. And these results are in agreement with the PCR result as well. We conclude that human polyomavirus 7, Merkel cell, and human polyomavirus 6 infect cholangiocarcinoma lesions, non-neoplastic bile duct, epithelial, and non-neoplastic hepatocyte. We conclude human polyomavirus 7, based on this chart, are more frequent to detect compared to Merkel cell polyomavirus or human polyomavirus 6, and all three human polyomaviruses are hepatotropic. Chapter 6, we perform the same techniques we use in Chapter 5 to detect and map the human polyomaviruses in renal cell carcinoma. We screen these 55 renal cell carcinoma by degenerated and by specific primers, as well by other molecular techniques, by FISH, RNA, and immunohistochemistry. Our results show some result for Merkel cell polyomavirus, human polyomavirus 6, 7, GC virus, and BK. We conclude in these chapters the frequent detection of human polyomavirus DNA and renal cell carcinoma suggests a potential reservoir for human polyomavirus latency in human. We conclude as well human polyomavirus DNA is frequently in the adjacent non-tumoral tissues. And finally, Merkel cell polyomavirus and human polyomavirus detection is higher than 6 and BK and GC virus. Finally, Chapter 7, which is a bit change. Here we're looking for the detections of bovine meat and milk factors in renal cell carcinoma. There are nutritional risk factor linked to human cancer. Meat and milk are the most popular food source in the most of the part of Western countries. Some groups show that an association among the food consumptions and human cancers due to this red meat or processing meat produce these chemical carcinogens. However, the other group shows not only the processing meat or the red meat can produce this chemical carcinogen, but also the white meat and fish. So, according to these studies, a new theory suggests adaptive rules of species-specific factors in the food consumptions and the development of certain disease. For example, based on this chart, it shows an epidemiological data among the food consumptions and colon cancer, and we see countries like Australia, Canada, and United States with a high consumption or high rate of food and high rates of colon cancer compared to other countries like Japan and Korea or India with a low consumption rate of food or red meat or processing meat and low rate of colon cancer. However, countries such as Japan and Korea, the rate sharply increased, and this is due to the meat imported from these Western countries on that time to this country. That's why they make the high rate of the colon increasing, while India, it can keep the same rate of colon cancer because the majority of people are vegetarian or they don't eat that much meat due to the religions as reported in that studies. What are BMMF? They are bovine meat and milk factors. They are circular single-stranded molecules around 1,000 to 3,000 base pair. They were isolated from cow milk, dairy product, and bovine, and due to the frequency detections from this dairy product named BMMFs, they were classified from BMMF1 to BMMF4 according to their molecular characteristic, and recently BMMF DNA have been identified in the adjacent non-tumoral of human colon, breast, and other cancer. Here we aim to investigate the prevalence of BMMF DNA in renal cell carcinoma and adjacent non-tumoral renal tissues. We use two collections by using this either degenerated PCR or specific PCRs. And this chart shows that BMMF1 and BMMF2 are present in renal cell carcinoma. And also this chart can show that the BMMF's DNA is high detection in the adjacent tumor tissues compared to the tumor tissues. We also use other 7 specific primers to isolate or to characterize the whole genome of BMMF, in this case in BMMF2. And this map shows our isolate in renal cell carcinoma, and this is in line with the previous detections in the dairy products. We conclude our PCR approaches for BMMF and BMMF2 DNA detection is a suitable tool to screen large formalin-fixed paraffin-embedded tissue collections. BMMF DNA are more frequently found in non-tumoral tissues. Our findings are in line with proposed models for BMMF-induced indirect colon carcinogenesis in adjacent non-tumoral tissues. Future studies need to address whether the described BMMF in renal cell carcinoma tissues are transcriptionally or translationally active. My conclusions: human polyomavirus and bovine meat milk factor are potentially cancer-causing agents. Human polyomavirus 6 were frequently detected in skin cancer, while 7 in non-skin cancer. Bika virus reactivation is not restricted to immunosuppression. Merkel cell polyomavirus and GC are present in urothelial cell carcinoma. Human polyomavirus 7, 6, and Merkel cell polyomavirus are hepatotropic virus. Human polyomavirus DNA in renal tissues suggest a potential reservoir for human polyomavirus latency or passenger in humans. And finally, BMMF DNA is more frequently found in non-tumoral renal tissues, and this is in line with the previous studies. Thank you for your attentions, and I would like to thank my team members and supervisors, and now I give the word to the prorectors.
Thank you, Mr. Mobaraki, for your very clear presentation. The opposition will be opened by Professor Tilanes, who was member of the assessment committee, but is also now secretary of the degree committee. He's a professor in transplantation immunology.
Thank you, Rector. Mr. Candidate, first I'd like to congratulate you with your thesis, it's well done, but I also like to congratulate the promoters and co-promoters guiding you to the interesting topics of this thesis. And also thank you for your presentation, but because that already raises me the one question, you mentioned that there are 4 different BMF factors. And so what are the you indicated that there are differences, so what are the main molecular differences between the 4, and why have you chosen 1 and 2 and not 3 and 4?
Highly esteemed opponent, thank you for your kind words and questions. Yeah, I mentioned that BMMFs were classified from BMMF1 to BMMF4 based on the molecular characteristics, and that because some of these BMMF1 were isolated from multiple sclerosis and other dairy products, and they have the same features of Sphinx 1.76, and others like BMMF2, they also have some similarities to Sphinx 2.76, which were isolated from TSE, transmissible spongiform encephalopathy, and the others from other multiple sclerosis or other dairy products, or and they also, it's not only the site of the isolation, but also the molecular characteristic to the some of these Psychobacters or Acinetobacter baumannii features. That's why the origins of BMMF is spanning between the bacteria and viruses.
So
and we choose, I'm sorry, to answer the other questions, we chose BMMF1 and BMMF2 because they were they were very studied, and they were detected in human cancer like colon and pancreatic and breast cancer.
okay, thank you. I want to move to chapter 7. And in chapter 7, I'm getting a bit confused on your PCR strategy. You described that you developed a highly sensitive, novel, broad-spectrum range PCR for BMMF1 and 2, but also, as you also showed, a consensus, and you have specific PCRs. So can you explain to me the strategy for PCR and identifying those factors?
Yeah, thank you for your questions. Actually, at the beginning, we designed these consensus primers, which were designed from the same conserved regions of these isolates, BMMF1 and BMMF2.
So they amplified together?
Yeah, not together. So because BMMF1 and BMMF2, they are different groups. So we just do an alignment for these isolates from BMMF1, and we designed this degenerative primer, and as well we do it for BMMF2.
Yeah, but sorry to interrupt you, but if I think it is a consensus, then they amplify both. So you have developed specific PCRs for BMF1 and 2?
I think, yeah, it's consensus PCR. I can call it, yeah, it's specific PCR for BMMF1 isolates. And the consensus PCR is specific for the BMMF2 isolates. On the other hand, we design other 7 primers to characterize the whole genome. So beside based on the consensus results, we start to also isolate the other part of the BMMFs.
So if I move to your results on page 156, you have there the agarose gel or the gels with PCR fragments. And in figure 1b, that is factor 1, you see the positive control, and I see many positives, but I also see other fragments. So what are the other fragments?
So, yeah, because we also construct this positive control, which has this his tag, so it should be a little bit higher than the positive results. So, yeah, so in case in 1C, yeah, it's only one band. I think that's, yeah, the bands we do a sequence when we expect it positive in this.
But in 1B, there are multiple ones at different locations. Have you isolated?
If it has multiple bands, we isolate actually the bands and we do it indirect.
And have you sequenced those?
Yes.
Okay.
The result, yeah.
And that confirms that they are factor 1?
By sequence, by Sanger sequence, yeah.
Okay, and but in the text you are describing that the frequency of factor 2, the C, is much higher, and then in non-tumor tissue compared to factor 1 that is displayed in figure 1b. And then you conclude, I have to have specific primers for the factor 2 and sequence the entire genome.
Yes.
Why haven't you done that with factor 1?
Yeah, it's actually they were BMMF2 were higher than BMMF1, and based on the frequency of detections. So we start to design these primers for BMMF2 because the high detections of.
But if I look at the figure 1b, which is factor 1, has much more positive ones than 1c.
That's true. I think, yeah, we have to change the gels. So we have some gels, it has more than, expected results.
Okay. I think first I give the word back to you, and if necessary, I can come back on the mechanism. That's okay?
Thank you very much.
Then I give back the word to the prorector.
Thank you very much, Professor Tilanes. And then the opposition will be continued by Professor Moons. He is a professor in molecular inflammation from the Arctic University of Norway in Tromsø. Welcome to Maastricht.
Thank you. First of all, I want to thank the university for the kind invitation and the kind hospitality. And I also want to congratulate the candidate and the supervisors and your team with the splendid work. I will focus on chapter 6. And my first question is an easy one. You said this paper is going to be submitted. Has it been submitted or?
Not yet, but I hope next week, because we have already invitations for this.
Okay. I ask because there are some mistakes, and one, for example, is in the abstract where you write that 40 out of 55 patients are positive, while in the discussion you write 44. And I don't think it's 55 patients, but 11 patients and 5 samples of each patient. So what is the exact number that are positive? Is it 40 or 44? So I think you should check that and make it clear.
Highly esteemed point. Thank you for questions. Yeah, but they are actually 11 patients, but they were cut from 5 sites, from tumor core and from transitional tumor and from other distance to the tumor core. But I think, yeah, in total they are 55, but based on the degenerative primer and combined with the specific primers, I think it's 40 out of 55. So is that a combination among those two approaches of PCRs? But I have to check, yeah, again to make sure they are in line with the other results in the context, in the main text.
Okay. According to my, but we can discuss later, according to my calculations, it's 43 that are positive, but anyway. First of all, you find that some samples are positive for multiple, simultaneously infected for multiple polyomaviruses. How can you be sure that it's the same cell that is simultaneously infected by two or several polyomaviruses? And it's, of course, you extract DNA from a sample which contains many cells.
Thank you for your question. It's really interesting, you know, to go further, to think about this point. Yeah, we actually just know that the same patient has the co-infections or simultaneously more than one polyomaviruses, but it's very interesting to look back to the cells and know how, if one cells, you know, includes two viruses or just the entire specimen or entire tissues.
How would you test that, if it's a single cell that has several, or if it's different cells in the sample that is?
So I think if, yeah, I think if by doing the in situ hybridizations like fish, we only see these signals for the DNA, but if we go furthermore to check by other techniques, which is actually I don't know, but I think there is another techniques to separate or sorting these cells based on this human polyomavirus distributions in the tissues.
You have two different antibodies, one for large the antigen of Merkel cell and one which picks up the other ones. So if you have a co-infection of Merkel cell and another one, you could do a double staining, for example.
A double staining, that's what's top 10. Her paper, she's doing these cocktails of these antibodies and screening some of these human cancers, but she states in the paper that doing the cocktail of these antibodies and screening at the same times, it gives you actually a strong signals than the using only one or two antibodies on the same times. But I don't know if you can see these patterns of the staining, which tells these two viruses contributes in the same cells. But I will think about these antibodies. And
what is nice that you have tested tumor tissue, but also adjacent tissue. Have you looked at the genome copy in the different regions, compared, for example, genome copies in tumor tissue and in non-tumor adjacent tissue? And if not, what method could you use?
We haven't analyzed that, but it will be nice to add it to the paper, actually, to give a strong opinion for these viruses, you know, contributes based on the genotypes of the virus, if they are heterogeneous or homogeneous. And
which method would you use to address this?
Just by Sanger sequence and blast and see the
genome, the number of copies.
The number of copies, yeah, we can do a qPCR to check that, but based on our results, the immunohistochemistry, we don't see these patterns in every single cell. So it's nice to do a qPCR to check the viral loads in the tissues.
Okay. If I still have more time, what was a bit puzzling for me is if you look at table 1 on page 132, with the consensus PCR, you pick up, for example, samples which are positive for Merkel cell polyomavirus. But when you use specific primers, 3 different sets of primers for Merkel cell polyomavirus, then the sample is negative. For example, sample 8, consensus PCR is Merkel cell positive, but it's negative with all 3 Merkel cell polyomavirus primers. Do you have an explanation for this?
It's well-known phenomenon that we have this result with the degenerative primer, but we don't have it for the specific primer. I know if the virus exists, it should be all positive because we have this primer as well for M1, M2, which targeting the large T antigens. But there are some speculations about this, but they haven't yet proved if the virus just exists and do the tumors and then it's escaped or left and only just a part of the virus in there. That's what they call it hit and run, but it hasn't been proved yet. So, however, the other primers, the targeting, for example, VB1, it's targeted the other regions from the virus, is not only, because the degenerative primer is designed from the large T antigen, and VB is designed from the late antigen. So I think, yeah.
If you find with your consensus PCR a product and you determine it as Merkel cell polyomavirus, why do you say that? Is it because of the size of the PCR product, or have you really sequenced the product?
It's by Sanger sequence. All these results proved by the Sanger sequence.
And that's why your primers have this M13 forward and reverse sequence?
Yes, for enhancing the sequence.
That was a bit unclear in the materials and methods. There is also some discrepancy between the immunohistochemistry and the PCR. PCR is more sensitive than immunohistochemistry, but you get some negative samples, PCR negative, but they stain positive with antibodies.
Yes, for these antibodies, like BAB 416 is for the it's an antibodies can use for different human polyomaviruses. However, some of these results we have it for the immunohistochemistry, but negative by PCR. We thought that it has a cross reactivity with other cellular epitopes or antigens, or it might be a novel polyomaviruses, which is haven't yet been identified yet.
Okay. Thank you.
Thank you very much, Professor Moens. The opposition will be continued by Professor Agul, who is professor in virology at the Köln Universitätsklinik. Welcome to Maastricht.
Thank you very much. Dear candidate, let's speculate. In your introduction, you mentioned that 15 polyomaviruses are known, and on your slides, you added a very recently identified one. We have now 16. Do you think that there are more polyomaviruses to come?
Highly stimulating. Thank you for the questions and kind words. Yeah, that's our purpose in this lab. Actually, we're using this degenerative primer and other immunohistochemistry and single cell level detections to see if there is other human polyomaviruses or novel human polyomaviruses. Yeah, but yeah, I mentioned it in my book. There are 15s, but recently some of these viruses were excluded by the International Committee taxonomy of virus because they were isolated from human, but they later found it in animal as well.
Yeah. And for the future experiments, which type of tissue do we need to test to find new polyomaviruses? So which tissues have not been tested for polyomaviruses so far? Do you have an idea or an overview which would be interesting to be tested? Yeah, we know skin.
As you know, I have read all of this publication, and they've been testing all the specimen from the brain tissues, cerebrospinal fluids, blood, skin. In our lab, we do liver, kidney, and bladders. So skin is very common, because these viruses shed actually from skin to other organs. So. Maybe, I think, if I think about it for future plan, I will do some screening for eyes, tissues, or to see how these viruses can play a role in the route of transmission.
Yeah. All right. For the polyomaviruses 6 and 7, you mentioned also on your slide there's a seroprevalence in the population is high and we get infected soon after birth. And in terms of evolution for humans, may they also have a beneficial role for humans? Maybe by inducing kind of immune challenge, a chronic immune challenge would be beneficial for humans, not causing harm, but be beneficial for us?
I think, so, yeah, thank you for the question. I think, yeah, that will help to improve, yeah. But in this human polyomavirus seroprevalence increasing with the age due to this autoimmune disease or immunosuppression or during the pregnancy.
Right. I want to ask a question to chapter 7, to the bovine meat and milk factors. So what are the missing links, actually, missing links to provide more evidence that they are playing a direct or indirect role in cancer development? So if you would have, let's say, €1 million for the next 5 years for research, which kind of experiment would you design to ascertain the role of BMMFs in cancer development?
Thank you very much.
Maybe even using artificial intelligence and all the new techniques which we can use.
Yeah, but it's very interesting because this other, from the other groups, they check for these replications in the tissue of the colon, and they don't only prove the presence of this replication protein of BMMFs, but also they do other staining to detect the immune response to this BMMFs in the colon by doing CD68 to see these macrophages, and they see the protein inside these macrophages, and they do other staining to detect if they create an inflammations, and they do other markers to see the ROS in this lamina propria of the colon. But yeah, in my term, yeah, if I will invest my research in future, I will focus on the immunohistochemistry to see the proteins of BMMFs, and also to do other cell lines with the transfection to see the not only the transcription but also the translation level.
I mean, there's so many data, or bioinformatical data on sequencing of all different kind of tumors. Would it be possible to extract the data for these factors from the data sets?
Yeah, that's where recently in our group we use pipeline to check these viruses if they play roles in other human cancers. So next gene sequence or bioinformatics to check, just collect this sequence and throw it to this database and see how they connect it to other human cancers.
Okay, thank you very much.
Another question.
So far, I'm fine, thank you.
Thank you very much, Professor Akul. Then the opposition will be continued by Professor Ramakers, who is Emeritus Professor of Molecular Biology, Genetics and Cell Biology from the Maastricht University.
Thank you, Mr. Prorector. Dear candidate, first of all, congratulations for the data of your thesis, also to your supporting team. I saw that you have a lot of data on polyomavirus, and there's one chapter that intrigued me very much. That's on these bovine meat and milk factors. And so I would like to extend a little bit on the questions of the foregoing opponent. I was just wondering, to get it clear, you have sequences of these single-strand circular DNAs. What are these exactly coding for? What are the proteins that are coded by these circular DNAs, finally? What are they coding for? You mentioned proteins that are responsible or needed for the replication. Other proteins that can be identified from these sequences that you obtained?
Highly esteemed point, and thank you for your questions. Yeah, it's actually this isolate has not only one open reading frame. Some of them they have more than 3 open reading frame, but the large one which contain the replication regions, and some of them have capsid protein.
Capsid proteins.
Yeah.
So the single-strand DNA, circular DNAs are included into certain structures, let's say protein structures,
Yep.
and then in fact, are taken up by macrophages. That's what I understand from your data, from what you told us, and also from, let's say, recent publications from the Zuhausen group in Heidelberg. So what happens then? Are they replicated in the macrophages? Or where does the replication take place if they do not infect the tumor cells themselves directly? Because the effect of these BMMFs is indirect, I understand.
I think, yeah, the only, as far I know, it's the replication's been tested with the human embryonic kidney tissues. Human embryonic kidney cell lines.
Well, that's in vitro, eh?
That's in vitro,
Yeah.
yeah. But when they tested for these colon models, they just prove the presence of this BMMF by targeting this replication.
Yeah. I'm just wondering how, let's say, if we
they designed,
these,
I think, 12 antibodies to just target this replication and show it in the tissue.
Yeah,
to show
because
that's
it's
the
not like
BMMF.
HPV. It's not replicated in the let's say tumor cells themselves. It's apparently present in the macrophages, and in as such, let's say, because of the effect inside the macrophages, the reactive oxygen species are produced, and those reactive oxygen species then induce changes in surrounding epithelial cells, for example, which then cause cancer. So I was wondering the routing of these BMMFs.
Yeah, it's actually basically the proof the inflammations by the presence of this BMMFs. One actually inside the macrophages.
Yeah,
That's one things to know. That's there is an immune response. The other things the proof that they create an inflammation by production of this radical oxygen species or nitrogen species by using another marker called 5XOXO, I think. Yeah, it's a biomarker to check the DNA actually mutation by productions of radical oxygen or nitrogen species.
Yeah. Okay, so far the routing. Then, let's say, based on the structure, you can actually identify a kind of a protein structure that would also allow you to make antibodies to these, let's say, the proteins that are produced by the single-strand BMMF sequences. The Harald Zuhausen group in Heidelberg has produced antibodies to certain subtypes and showed very nicely. I mean, from your sequences, could you deduce certain protein sequences that could be used for the production of antibodies?
Actually, that's our future plan to check these BMMFs, and not only in the transcriptional but also in the translational level by
really
to this sequence and
targeting.
targeting.
Because I mean, DNA can be present. Okay, sorry to interrupt you. If I have some more time, Mr. Prorector. So we are talking about protein, let's say BMMFs, which produce protein also. Can you detect these? Do you think you can detect these BMMF circular sequences, but also their proteins in the bloodstream? It has been done, probably.
There is a paper in 2008 done by Sebastian Elbrecht in Germany. He tested the health blood donors for the presence of these anti-replications or replication protein for BMMF by using ELISA, and also they were positive, and he used another neutralizing ELISA to specify or to make it specific to this replication regions by using specific monoclonal antibodies.
So that is a protein, but what about the DNA? Can that be detected in the blood?
Not yet, but
No.
we found it in the renal cell carcinoma, in the tissue, and
yeah,
the
you
others
saw
in the
it,
colon. And but also, it's actually BMMF were isolated from the brain and from the of multiple sclerosis
Yeah.
tissues, but also I think in blood as well.
From those, let's say, patients that isolated the DNA and not the protein.
DNA, yeah.
Yeah, yeah. And
The
final
whole
question,
sequence.
maybe, let's say, in case it turns out that such BMMFs seem to play an important role in many types of cancers, would it be possible to produce vaccines, for example, against these BMMFs?
Yeah, that's what Harold mentions early when he talks about the speculations, and he mentions about first for long breastfeeding, more than 6 months, because this milk of mother has some sugars which can bind to receptors of the host cells and block the binding to the BMMFs. The other things he talks about this vaccine by targeting the capsid protein or the duplication proteins to prevent the BMMFs replications or infection or increasing the inflammations. And the last things he mentioned about this non-steroid anti-inflammatory drugs, which have been studied in the diseases with the inflammations to reduce the inflammation.
And another option, let's say, to prevent the effect of these BMMFs in tissues is to scavenge away the reactive oxygen species. So let's say oxygen species scavengers would also help in preventing these cancers. I thank you for your answers and give the word back to the project. Thank you.
Thank you, Professor Ramakers. The opposition will be continued by Professor Stickeler, who is professor in obstetrics and gynecology and chairman of the department in the University Hospital Aachen.
Thank you, Mr. Prorector. Dear candidate, I would like to thank you for your work and also congratulate on your team and the supervisors. My first question would be more a general one. It's going back to your proposition number 8, stating that global cancer incidence linked to infection will be increased over time. Could you reflect a little bit in regards to your work? How would you put this in perspective? Do you have any underlying hypothesis for these proposition and maybe different kind of cancers which you would like to focus on, and what would be maybe a major health perspective in the world?
Highly esteemed opponent, thank you for your kind words and questions. So you mean it's proposition 8?
Correct.
Yeah, actually the speculations of BMMF start from studying this epidemiological data from this aspect and link it to the increasing of the cancers. So I think thinking about the observations from the epidemiology data will help to increase the insight or the pathogens novel in futures.
So you focus now on BMF1 and 2 in your answer, but I think this was a more general question in regards to different kinds of virus types, like your polyomaviruses also.
I think, yeah, I remember one of this paper from Carolina Torres. She was talking about this evaluation of the polyomavirus based on the countries and geographic area, and they found some of these polyomaviruses in countries like Africa or Europe, but they are less in other countries. So I think the geographic distribution play around in this polyomaviruses, but also there is another paper from Laura Manuel Des. She's discovering this Sphinx 1.76 and 2.36, which is an infectious agent, and she was looking for these geographic distributions of the animals and link it to the environment. And it was thought actually this TSE, it comes from the prion proteins transform itself to be infectious agents. But later she studied the environment and she found there is actually other infectious agents. And they when reduce these infectious agents, a disease like cow mad, which is bovine spongiform encephalopathy, it's actually decreased. And other diseases like Koro, which in human brain, because these people eat the brain of tissues, and when they reduce the consumptions of the food based on that geographic area, it helps to reduce the disease. So thinking by the epidemiological and cancer really helps to preventions or to discover new diseases or new pathogens.
Okay, thank you. Then I would like to focus back on BMF1 and 2. You mentioned that there are data showing that you find it in the surrounding tissue of breast cancer, for example. Have you any explanation why you have a specific organ tropism, like you showed data now on renal cell cancer, you showed on breast cancer, on your cholangiocellular cancer? So is this just picked out now by chance, or do we have really a tropism, organ tropism for these infections?
Thank you for your questions. The speculation starts from Harold Zorhausen, that he was thinking about the most common cancers in human, in male, and the most common cancers in female, and colon is one of the most, male cancers, and breast, the other in the female. And he looks for these food consumptions in these countries and connected, because this kind of cancer was connected with the inflammations, and he found out that there is a relationship between these diseases, not only because the chemical carcinogens, but because there are other species which are involved in this human diseases. For the BMF1 and BMF, we start with the screening them because they've been already detected in cancer like colon cancer.
So, but are there known clear mechanisms and maybe differences between epithelial cells to take in these BMF1 and 2 molecules, for example? Is this known?
Thank you for your questions, but as far I know, and when I read these papers, actually it has the same receptors on the host cells, like sialic acid or other receptors which can bind to these molecules. That's why he mentions that if we long the breastfeeding, which makes these sugars bind to these receptors and block the receptor to be bind to the BMFs, which helps for reduce the infections of these infectious agents.
Okay, do I have one more question? Time? Okay, so one more question now in regards to carcinogenesis. So in your book you proposed, or you showed the two different pathways of carcinogenesis, indirect and direct. If we talk about, we've heard about indirect in regards to the oxygen stress, but in direct ways, do you have any data on the integration of the BMF1 and 2 into the host cell DNA? Is there any kind of hint for this?
We haven't checked for that, but it will be interesting if we do next gene sequence for our cohort and see if it's integrated or it's in there.
So this was not done yet?
But it's better to do others like fish and to see if it's, and then we can confirm our result. If the fish is positive or the RNA is positive or protein is positive, they do the next gene sequence to confirm it's integrated to the host DNA.
Okay, thank you. So thank you very much for your answers, and I give back the word to the prorector.
Thank you very much, Professor Stigler. The opposition will now be continued by Dr. Severens, who is a pathologist working at the Department of Pathology at the Maastricht University.
Mine is missing. So, dear candidate, first of all, I would like to congratulate you and your team on your hard work on this thesis. I really enjoyed reading it, and I'm impressed by your results. I think it's really fascinating that a group of viruses that gives so few clinical symptoms that can be involved in carcinogenesis. So my first question, I would like to start easy, and is related on proposition number 9. Maybe you can read it out loud for me.
Highly stable point. Thank you for your kind words and questions. High frustrating tolerance is an essential requirement for PhD students.
Yes. So I'm wondering about your personal experience and opinion about that.
Actually, to be independent PhD students and take care of everything from the scratch, you know, since you receive the data, the blocks, the tissues, you start the experiment, you start your manuscript, you submit it. It's a long process, but sometimes we face a lot of obstacles, and we encounter some problem with the techniques, and we have to deal with it and fix it. And you have also deadlines, so because you need to submit this result to start, you know, the next step. So it's frustrating, yeah. But if you think about how to be patient and tolerance beside the frustrating, and think more, so we can help to fix these techniques and back on the same track and continue. But actually, where I get frustrating in my PhD journey this time, because of the coronavirus and everything locked down at that time, even not easy for the shipping or ordering the reagents and anymore, and we have to be restricted to the rules on the lab, and yeah, it's difficult on that times a little bit, but after the lockdown, we back at least to the same track, and we continue working and finishing and finalize our results. But I'm not frustrated anymore.
That's very good to hear. Okay, so next I would like to take you to chapter 3, in which you investigated the presence of BK polyomavirus in urine cytology specimens. And as you explained in your presentation as well, you had a total of nearly 23,000 urine cytology samples from our department over a period of 15 years. And you found 30 cases with a BK virus infection. Could you explain to me how you selected those cases?
Actually, the analysis of these specimen cytology cohort, it was analyzed by very smart and intelligent pathologists, and she helped me actually to sort out of this patient based on the clinical data. And the presence of BK virus, it was not reported on that database. We just choose the ones it has the decoy cells, and based on the decoy cells, we say it might be a viral infections, either BKGC or other viruses like say Ghalib, for example. And then I receive the blocks and I start the experiment. But the analysis of this was done by the pathologist in our department.
Yes. And did you select cases with decoy cells, or did you select cases with the SV40 staining? Because did all cases have the SV40 staining already in our archives?
I think both, yeah. By the Papa Nicolau staining and SV40 in the same time. But we, it was for the urine sediments, but we screened the other FFP tissues by applying this SV40 again to compare to these patients, not only in the urine sediment, but also in the tissues.
Yes, I think you might have missed a couple of cases because we don't standardly perform an SV40 staining, but we report them as decoy cells. You showed a beautiful picture of it in your presentation, and also in your thesis. So we don't always perform an SV40 staining. Okay, so my next question is also related to the decoy cells because you did not find SP40 staining in the urothelial cell carcinoma and the paraffin-embedded tissues. So I was wondering, did you think about the origin of the decoy cells? Because, well, urine is produced in the kidney and excreted by the kidneys and stored in the bladder. So perhaps the decoy cells are not of urothelial origin, but more like tubular epithelium origin. Have you considered?
I haven't, think about this from this point, but that will help actually to sort again this specimen and see if there is a relationship. Why we don't have it in the tissue, but we have it in the urine, and that's maybe the point because it's not originated only from the urothelial, but other trabecular tissues.
Perhaps I can explain why you didn't find a relationship. Yes. Okay, so I have another question for you. This one is about chapter 5, in which you investigated several polyomaviruses in cholangiocarcinoma cases. You investigated virus 6, 7, and Merkel cell polyomavirus, but you didn't investigate the GC polyomavirus in this study. And I was wondering why not.
It's actually, when we start with these degenerative primers to fast screen in every tumor to see how the presence of these viruses in our cohort. And based on the degenerative primer, we apply for specific primers to confirm our results. However, it will be better if we screen the cholangiocarcinoma patients with these big angiosophiles to see if they are.
Finish your answer, or you were already?
So maybe in futures we apply this PCR to see how they play around in the cholangiocarcinoma patients. Will be interesting. Thank you for questions.
Mr. Ghalib Mobaraki, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. I request that you and your company await the results of our deliberations and our return in this room.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. All your time, 'cause I'll go the extra mile. Long road to the south side, 7 miles in my rearview mirror, I know what it felt like. My goal's only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and die. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Hit the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get everything I ever hoped for. It's a challenge and we all in. It's a better future that we go for. Even though these nights seem long, I know that I am never on my own. Know you've won the battle once you've grown. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and that. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and that. What's on the line? I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. 'Cause I'll go the extra mile.
الشهادة.
Ghalib Mobaraki. The degree committee here present has assessed the quality of your thesis and your defense. In view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Zurhausen is authorized to confer you upon your, this academic distinction in accordance with the Dutch university custom. I invite your supervisor to now take the floor.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
I promise.
By the authority vested in us by law and in the conformity with the decision of the committee here present, I hereby confer upon you, Ghalib Ibrahim Mobaraki, the degree of doctor and grant you all rights attached by the custom and law. As evidence of this, I now present you with a degree certificate signed by the rector, the secretary, and the other members of the committee and affixed with the official seal of the university. Surprise. Dear Dr. Ghalib, dear Dr. Mobaraki, congratulations. That was really an impressive presentation. It was an impressive discussion, and I think it might not occur as a surprise to you, but you still remain a big surprise to me, so as I might have indicated before. So I'm very impressed about your achievements during the last 5 years. You came here 5 years ago. Basically, you said you were coming, but you didn't. And it took some extra months to be here then, whatever the reasons were, but suddenly you were there. And then it is already for us Germans, it's sometimes difficult to work in the Netherlands to understand the culture, which we think is so close to us. But how must it be for someone, or how is it for someone from the other cultural surrounding coming here to the Netherlands? And indeed, we, as an international team in our department, also in our group, there were some, let's call it cultural pitfalls or clashes, so which you really mastered to overcome all these problems. And so you started your work here. And as you know, it took me some time to understand you, to follow your approach, your personal approach to science. And it took me some time to understand that this is also a very valuable approach to address problems and topics and how to solve problems in the laboratory, especially technical. And you're really strong on the technical floor. You really are into experiments. You're also very much into teaching. So it's a great achievement which you show in total in this 5 years. You mentioned already in your talk that you had the last 5 years. There was an incision in this time by COVID, which really took lots of time from you. The laboratory was closed. And when they reopened, there were no consumables anymore because everything went to microbiology department for the COVID testing. So that was really took us a long time, and so we were happy in first instance that your contract was prolonged by university. And unfortunately not in this period as we'd like to, so that's why we're here already today. But so you managed this as well, and you managed the discussion here in a very sovereign way. So I'm very happy with this achievement. So basically now you will be turning back to Saudi Arabia. And I think at the beginning of next year, you start your position at Jazan University. And I'd like to personally address the representatives of the Jazan University, if they are somewhere listening, just my recommendation for this young and dedicated scientist, because I think that you will have a bright future if you carry on your work as you do this, and you're very down to the point. And so I think that we will also cooperate in the future, as we already do with your previous colleague from Saudi Arabia, Faisal Klufa. So I think this will be very prosperous cooperation. I hope so. And I wish for you and your family the best for your private life, but also for you, for your professional life. And no need to say that I'm very proud of you, really.
Thank you.
Congratulations.
Thank you.
Dear Dr. Mobaraki, also on behalf of Maastricht University, we congratulate you with the degree you have acquired. We were quite impressed today with your defense. I think you did excellent, and I hope you will leave all the frustrations behind here in the Netherlands. You will return to your country, and well, good luck with your scientific career, and also good return to your family in Saudi Arabia. Hereby we will then, I have to thank the opponents from outside of Maastricht, Professor Moens from Tromsø, Professor Agul and Professor Stikerov from respectively Cologne and Aachen. Thank you for coming to Maastricht to take part in the defense ceremony also. Now we will leave this room. We go for the stairs to take some pictures, and the audience can go to the rafter for the reception procedure. And hereby I close this academic session.